Hello everyone. Today we are going to discuss about gene cloning. This is the outline. What is gene cloning? Requirements, steps involved in gene cloning, advantages and their limitations. First we have to know about what is gene cloning. Gene cloning is a molecular biology technique to create exact copies of particular gene, that is our gene of interest. It is also known as molecular cloning or DNA cloning or rDNA technology. Then requirements: DNA fragment which contains the desired gene. Restriction enzyme. It is used to cut the DNA fragment. It is also known as molecular scissors. Ligase enzyme which helps to join the ends of DNA strands. So it is also known as genetic glue. Then vectors. Vectors are carriers to carry the desired gene, and host cells. It is used to replicate the gene. Next, steps involved in gene cloning. Step 1. Isolation of gene of interest. We have to isolate the DNA fragment, that is desired gene, by using restriction enzyme at the restriction site. Step 2. Selection of vector. The vector must be self-replicating, unique restriction site, easily isolated, and it contains marker genes. The vectors are various types. It may be a plasmid, bacteriophages, cosmids, bacterial artificial chromosome, yeast artificial chromosome, and mammalian artificial chromosome, and etc. Step 3 is the formation of recombinant DNA. The desired gene is inserted into the vector by cutting the vector. Here the important thing is we have to use the restriction enzyme which is used for the isolation of DNA fragment in step 1 and paste it with the DNA ligase. The new DNA is formed. It is known as recombinant DNA. Step 4. Transformation of recombinant into host cell. The transformation occurs naturally in some bacteria like Helicobacter pylori. Some bacteria requires chemical methods like electroporation, microinjections, liposuctions, and so on. Step 5. Isolation of recombinant cells. We are already transformed recombinant DNA into the host cell by transformation, but some of them only intake the recombinant DNA, so we have to isolate it from non-recombinant cells using marker genes of the vector. These marker genes helps to separate the recombinant cells from non-recombinant cells. Step 6. Multiplication of selected cells. Once the recombinant cells are separated, multiply the cells in culture media to replicate the more number of copies of DNA fragment. Step 7. Isolation and purification of the product. Isolate the gene of interest and purify the product to further use and store. Next is advantages. Gene cloning is used to study the gene function, gene structure, and also used to construct the genetically modified organisms, and also is used in gene therapy to treat genetic diseases. Moreover, used in biopharmaceuticals for the production of recombinant proteins and etc. The limitations are it is very expensive and time consuming process. There is not sufficient yield and accuracy and very laborious process. Thank you.
Hi everyone, I am Jay Shruti from the Department of Biotechnology. Today I am going to share about microbial production of vitamin B12 and B. Steps of vitamin B12 production that I include fermentation, recovery, purification and functions. Vitamin B12 is also known as cyanocobalamin. It is water soluble which plays a vital role in the normal functioning of the brain and nervous system as well as in the formation of blood in animals. Sources of vitamin B12. It is mainly obtained from microorganisms. Human it is obtained from meat, fish, dairy products, and ruminant can obtain from the food web. Daily requirements of vitamin B12. It is daily required about 2.4 microgram and higher amount is needed for pregnant ladies. Production of vitamin B12. Microorganisms such as Propinium bacterium and Pseudomonas species which can be achieved to 300 mg per liters. For whole fermentation purpose, bacteria are oxidated for 2 to 3 days at 30 degrees Celsius. The salts needed for vitamin B12 include cobalt ions and related minerals, and the precursors that involved for vitamin B12, choline and betaine. Then it extracted by 80 to 120 degrees Celsius for 2 to 3 hours and the pH requirement 6.5 to 8. Product recovery of vitamin B12. The steps include extraction, membrane filtration, absorption. In downstream processing, the steps involves cell mass concentration, drying, centrifugation. Potassium cyanate is added along with the sodium nitrate followed by heating, then purification. Vitamin B12 is treated with zinc hydroxide to purify. There are a number of factors that affect the degrees of purification and then crystallized by addition of organic acid. Importance of vitamin B12. It is required for function of central nervous system and to produce healthy red blood cells DNA synthesis. It also helps the nervous system and the immune system to run smoothly. It also play a crucial role in skin. Microbial production of bee involves types of bee, steps of bee production, fermentation, packaging, health benefits. Firstly, beer is a fermented beverage produced from malt by a process called brewing. Types of beer. First one, lager. It settles down at the bottom of the vessel. Pilsner, colorless lager. Ale, it settles on the top of the vessel. Porter, it has highest alcohol concentration than lager. Stout, it is also called a black because of its color. Raw materials for the barley, adjuvants, tannin, water, yeast. Steps of beer production. First one, malting. Barley grains are first cleaned for malting, followed by dipping in water for 2 days. Then it incubated for 5 days for germination. Then kilning. Germinated seeds are fried at 80 degrees Celsius. So kilning is a fried process. Malting. Grains are crushed to create a coarse powder. Mashing. Mailing step is now kept in warm water for 1 hour. In this step, pH and temperature plays a very main role. Slightly increase or decrease in pH and temperature reduces the enzyme activity resulting and it leads to low quality product. Boiling of what? The raw product is the filtered water is boiled and then extracted by sugar caramelization. Then hops or dried flowers of Humanus lupinus is enough for 1 barrel of beer to provide additional flavor. Then the process called fermentation. Saccharomyces varum and Saccharomyces cerevisiae are utilized in the creation of beer. Beer fermentation generally happens at 3 to 4 degrees Celsius. The tank is open fermentation. The alcohol content of beer at large is 3.5 to 5 percentage. Packaging of beer is either in cage or glass bottle. The beer bottle is filled with counter pressure to reduce oxidation. Health benefits of beer. Xanthosumol, the flavored component in beer, plays a significant role to control prostate cancer. Its digestive system is it has many digestive qualities, including the activation of gastrin, gastric acid, and pancreatic enzyme. It also reduces risk of cardiovascular diseases by accumulation of an amino acid called homocysteine. It makes it as a good source for anemia prevention and also B reduces tension and it promotes sleep. Thank you.
Hello everyone, I am Darshini from Department of Biotechnology. Here I am going to explain the topic isolation and screening. Screening is the procedure of isolation, detection and separation of microorganisms of our interest. So the source is microorganism, food that includes milk, cheese, environment that include air, water, soil and compost and other sources. The important things that to be considered while screening is choice of source, choice of substrate, and choice of reduction. Screening is classified into primary screening and secondary screening. The first of primary screening is the isolation and reduction of a microorganism of our interest. So here it determines the organisms are capable to produce a compound. The first type of primary screening is organic acid producing microorganisms. The pH indicating dyes is used for detecting the organism capable of producing organic acids. So the dyes undergoes the color changes according to its pH. So the dyes are neutral red and bromothiamyl blue. So incorporation of calcium carbonate with a nutrient medium is used to screen the organic acid producing microbes. Second step is the antibiotic producing microorganisms. So here crowded plated technique is used. This does not give the information about the sensitivity of antibiotics towards other organisms. The purified cultures are then tested to find the microbial incubation spectrum that include first a spreader soil dilution on a plate of a selective medium, so then incubate, so then overlay with the integrated organism incubate. This gives the non-producing organism zone of growth, inhibition and producing microorganism. The third is the extracellular metabolite producing microorganism. This is an oxonotrophic technique, so that is able to produce a growth factor, vitamins, amino acids extracellularly. This includes two major steps, that is the preparation of first plate and second plate. The first plate, it is lifted and placed on a second plate without inverting. The growth factor produced in agar can diffuse into the lower layer of containing the test organism. The fourth technique is the enrichment culture technique. This was designed by the Beijering to isolate the designed microorganism from heterogeneous microbial population. It consists of the following steps, that is the nutrient broth is inoculated with microbial source meal and incubate. A small portion of inoculum is plated. So suspected colonies from the plate are subcultured on a fresh media. This is the isolating an organism from a natural sources. Next, secondary screening. Primary screening helps in the detection and isolation of microorganisms from natural sources. So, but it does not give the in detail of the production potential yield of organisms. So, such details are given by the secondary screening. So, this can provide broad range of information pertaining quality of the yield product, elimination of the contaminant, type of the fermentation product that is able to perform. So, and the product is toxic or not, yield of potentials, various isolated microorganisms, and so on. So first technique is Jane colony techniques. So this technique is used for the isolation and the detection of those antibiotics which diffuse through the solid medium streptomyces. The isolated streptomyces culture is inoculated into the central area of sterile plate. So the cultures of the test organism whose antibiotic sensitivity is to be measured are streaked from the edges of the placement. So then the distance over the growth of the different test organism is inhibited by the antibiotic secreted streptomyces is measured in millimeters. So this is the growth factors from the gypsum, incurrent and the filworm. So second is the filtration method. So this method is employed for testing those antibiotics which are poorly soluble in water or not diffused through the solid medium. So then liquid medium method. So this method is generally employed for the further screening to determine the exact amount of antibiotic produced by organisms like a streptomyces. So this is also aerated by shaking continuously and vigorously during the incubation period. So what kind of organisms are isolated during this secondary screenings are antibiotic producers, Cephalosporium acronyms, Penicillium chrysostom, Bacillus subtilis, Streptomyces crisis, Aspergillus niger, Candida tropicalis, secondary metabolite producers. Thank you.
Hello everyone, this is Jay Shruti from Department of Biotechnology. Today, let us going to explore the basics of shake flask fermentation. Introduction of shake flask fermentation. It is a laboratory scale and it is used to study about microbial growth and its production of our desired compound. The components that we need: flask, culture medium, and suitable microorganism, and shaking. That process is called agitation, that provides uniform suspension of cells in homogeneous nutrient medium. The importance of it, it is a simple and cost-effective model for understanding larger scale fermentation processes. The steps involved in shake flask fermentation. First one is the inoculation step. Introduce the microorganism into the flask and incubation. These cultures are grown under controlled conditions like pH, temperature, etc. The next process, agitation, is used to shaking the flask to ensure uniform mixing and also oxygen distribution. The periodic sampling is used for analyzing and monitoring. The benefits are it is very low cost and requires minimal equipment and resources. It is suitable for various microorganisms and various products. It is easily monitoring the sample and analyze culture parameters. It is rapid screening for evaluating multiple strains or conditions. The challenges of it is limitations that I already discussed. The cheese fermentation is limited in oxygen supply, so it is limited in scale, and also mass transfer issues is involved and the inconsistent oxygenation. The future prospects of it is advancement in bioreactor design. It is ease of operation and reduces material and capital expenditures, better throughput, and also reduce manpower requirements. Applications of shake flask fermentation that involve the first important step is strain improvement, that's selecting high yield microorganisms, and next the optimization of the product is optimal condition for the product formation is very important. Bioprocess development, that scaling up successful flask experiments to larger bioreactors, assessing the impact of pollutants on microbial communities, the continuous role in a rapid prototyping and also screening in bioprocess development is involved in the field of biotechnology. And thank you for watching.
Hi, hello guys. We have to discuss about the types of fermentation. First of all, we have to know about what is fermentation. The word fermentation is derived from Latin verb fervere, which means to boil. Fermentation is a metabolic process that converts sugar to acids, gases, or alcohol. It occurs in yeast and bacteria and also in oxygen starved muscle cells, as in the case of lactic acid and fermentation. Now, we have to discuss about the types of fermentation. There is a three types of fermentation, that is the submerged fermentation and solid state fermentation and a continuous fermentation. The submerged fermentation is nothing but we use a dissolved substrate in a suspended in large amounts of water or media. Examples are pickling vegetables, producing yogurt, beer, wine, and soya sauce. Subdivided into aerobic and anaerobic. Aerobic, citric acid production by Aspergillus niger, and anaerobic is yogurt. When submerged fermentation is done in flask with continuous shaking, then this is known as a shake flask fermentation. And solid state fermentation. In contrast to submerged fermentation, solid state fermentation is the cultivation of microorganisms under controlled conditions in the absence or nearly absence of free water. It has the characteristics, that is low moisture levels, smaller reactor volume, low contamination due to low moisture, easy product separation. Examples: mushroom cultivation, bread making, manufacture of miso, that means soya paste, soya cake, and cassava. Then finally, we have to discuss what is the continuous fermentation. In continuous fermentation, the fresh media is added to the fermenter continuously at a fixed rate. Spent media is also removed from fermenter. Both removal and additions are done at same rate, so the working volume remain constant. Under these conditions, the additions or removal, the cells remain in logarithmic or exponential phase of growth. Thank you guys.